So we'll just build on what Greg talked about here. Should point out that of the 17 sites we installed, we did this part of the study only on 8 of them. This is up here in the Olympics. We had a complete block up there, and the Willapa 2 block here, we had all 4 sites there, so 2 replicates of each treatment, including the references. And we did that because of 2 things. One was cost, but the other was it's just not possible or not logistically feasible to measure flow or to install flumes in a lot of these sites. So the objectives were to estimate the change in the export of nutrients, meaning nitrogen, a couple different forms. We did measure total nitrogen, ammonia, and nitrate nitrogen, and suspended sediment. And again, we did this in just 2 blocks, 8 sites total, had continuous stage height measurements here in the flumes, and we used that to calculate flow. And then at the sites, 2 sites where we had to use culverts because we couldn't find a place to install a flume in the stream, we just came up with our own stage height flow relationship there. And then we took periodic water quality measurements, actual water samples, and sent them in for chemistry. We did that at about 6 to 8 week intervals, so a lot of those would be during low flows. And we also wanted to get high flow events, and we did that by utilizing a method called turbidity threshold monitoring. It's developed by Jack Lewis and Rand Eads in Casper Creek in Northern California. And what that consists of is, so here's your data logger that controls everything, power source. This is a pump sampler. It's got 24 bottles in there, and it's got a piece of tubing running out here to the stream, and it runs down here in the stream. Inside of here is a turbidity sensor, and again, that would come on every 10 minutes and measure turbidity in the stream. These are actually quite sensitive. This thing was set up so that when flow comes up, it just kind of lifts that PVC pipe up, keeps the end of the sensor in the water, but lifts it up, and so any debris coming through usually just passes right on through and doesn't block things up. So during high flows, you can see how turbid it is. Okay, so this is our flume. We had this pressure transducer in here to get stage height. Then during high flows, when turbidity is up and stream stage is up, flows are up, it would trigger samples. Here is the pump sampler, or the turbidity sensor back here. Would trigger a sample, just pull it up out of the water and into the bottles. So what this ends up getting you is, instead of having, if you go out there at periodic intervals, flows are low most of the time, turbidities are low most of the time, and most of your samples are going to be down at that low end of the spectrum. What we wanted to do is sample all across that turbidity spectrum. And so this shows one storm event, with blue here being flow on the right axis, red is turbidity, and these dots are a sample collected. So as soon as flow starts going up, you get a little bump in turbidity. On these dashed lines here represent the thresholds that we had in the program. So when turbidity crosses one of these thresholds and flows above a certain stage, it will trigger a sample. Then it won't collect another until it crosses another threshold. And then it does this as it's going up, thresholds going up, and then as it crosses the thresholds coming back down. And so you get a complete sample of the storm, and you get a whole range of fairly uniform representation of the range of turbidities that are out there, instead of oversampling the low flows like you typically do. Then the sample, you would take those turbidity, those suspended sediment concentration measurements you got from your water sample, and you could regress those against turbidity and come up with a way to predict what suspended sediment concentration is based on turbidity at those 10-minute intervals. And then you calculate load simply by doing a concentration times flow for each 10-minute interval. So again, we used the first 2 years of pre-harvest data to build these regression models to estimate concentration. And we did this was originally developed for suspended sediment, but we also did the same thing with nitrate, total nitrogen, and with total phosphorus. And in the case of phosphorus, we used turbidity and flow as predictors, and in the case of nitrate, it was mainly flow. So we could estimate concentration each of those 10-minute intervals, estimate the load as concentration times load times time over that 10-minute interval, sum those 10-minute intervals into whatever time interval you want. So we did it into daily measures of export during that day. And then it gets a little complicated. So we've got export for each one of these, for phosphorus, nitrogen, and suspended sediment for every day. We've got these for about a 5-year period of time. And since export is really driven by flow, high flow events are going to transport more, whether it's nitrate or sediment. And so when we compare the unharvested streams to the reference streams, we've got to compare them across that same time interval. So what we did was all of our analyses were based on the date that basin was harvested. So the date harvest began, the year prior to that was the first year pre-harvest, the year prior to that was the second year pre-harvest. And that may be a different year than the basin next to it that was harvested on a different schedule. Likewise, post-harvest began once they finished the harvest. First year post-harvest was 1 year after that. And so we used calculated flow for each of these post-harvest years for each of the treatment basins, and then we compared that, or actually subtracted out the export from the reference basin for that same time period. So it's all comparing like flows to like flows, same time period to same time period. And then for analysis then, we took those differences between what we measured in the treatment basins pre and post and what we measured in the reference basins pre and post during that same time period and fed those into the mixed effects models and looked at fixed effects again were the buffer treatments in the pre and post time periods. So looking at the results, this is that same graph we saw earlier looking at suspended sediment. And this is a real typical pattern we saw. You'd get, this is over a like 30-hour time span early on in the study. But this was typical pre-harvest and post-harvest. As soon as flow would go up, you'd start getting bumps up in turbidity. Here, flow goes up, peaks, and as soon as the flow stops increasing, turbidity drops way down. Here again, you get a peak, and it's not that flows are dropping down very much, but turbidity drops down almost to background level. So it's very flashy. It's probably indicating a sediment-limited system, that as soon as flows goes up, the instant flows are up, they're transporting whatever that flow can transport. As soon as it decreases, there's not much there left to transport, and so turbidities drop. And what this amounted to was on a scale of 0 to 1,600 NTUs, over 90% of our measurements across all time periods, the entire study, and across all treatments, pre and post-harvest, were less than 1.4 NTUs. Meaning turbidity was low most of the time, both pre-harvest and post-harvest. Now that wouldn't be I think that tells us a lot in one sense, but the other thing that happened just logistically, or I guess more to the methods, is when we predicted suspended sediment from these turbidity measurements, we used a logarithmic scale. What happened then, and we're also measuring turbidity down at the low end of the scale, very near the limits of what we can measure. Likewise, suspended sediment concentration, we're measuring it at, or usually down at that end of the spectrum, we're down below what we can reliably measure in the lab. And so we're kind of pushing the limits of what we can do down in that area where 90% of our measurements were. And when you throw in the logarithmic scale we used in the model, what it did was predicted a non-zero concentration, even at very low turbidities. Now, it doesn't sound like very much. But if you're predicting, a milligram of suspended sediment at a time that covers 90% of your study, and you sum that up, then I think what we ended up getting was, estimates of sediment transport that were biased high simply because of this artifact in the model and the fact that turbidity was so low during the entire study. So bottom line is I don't think suspended sediment, based on all this, I think our estimates of suspended sediment in the study report are probably biased high because what it shows is most of that suspended sediment is being transported during low turbidity, low flow events, which is not very likely. So we'll go on to nitrogen here. A couple of things we learned is that first thing was kind of surprising is the pre-harvest concentrations varied widely. Now if you look in the literature, there's a lot of variability there, but our highest and our lowest, so 98 micrograms per liter flow-weighted concentration and 800 micrograms per liter flow-weighted concentration were in adjacent watersheds, which are, just to look at them, are identical in the species composition and almost everything. And so I'm not really sure what's driving that. So they are quite variable, and there's a third watershed that's adjacent to these two, which was somewhere in the middle. So there was a lot of variability in pre-harvest concentration. In spite of that, I mean, there's a uniform increase in nitrogen concentration. When I say nitrogen, it includes both nitrate and total nitrogen, because nitrate comprised the vast majority of the nitrogen we were measuring, and the results are almost identical. But concentration did increase post-harvest in all sites, and the increased concentration plus that increase in total runoff, total discharge that Greg was talking about, means that we did have increased export post-harvest. And again, most of the nitrogen is in the form of nitrate. So just to give you some examples here, in white are pre-harvest or concentration in the absence of harvest. So you can see some of the variability here, 98, 800, you know, that was the highest and lowest, and these are adjacent watersheds. And the Olympic sites are somewhere in between that. Likewise, pre to post-harvest changes, you go from 98 to 263, so 2.5-fold increase in concentration. This was maybe 10%. So the magnitude of the increases, however you express it, varied, but there were increases across the board. And if we look at export, we go from a low of 1.7 kilograms per hectare per year. That increased post-harvest to 7.26. And again, across the board, increases in export. Again, this was the watershed, this Olympic 100% had the smallest increase in export, but it also had the least area that was actually harvested. Only some 46% of that watershed was actually harvested. So looking at timing of this shows the harvested basins, these bars. The red is the harvested basin. Export, this is a pre-harvest period to the left of this dashed line. References in blue. I think the thing to focus on is the difference between these. So in this case, the harvested basin was slightly higher than the reference basin pre-harvest, except during extreme flow events. Sometimes it'd be much higher. You go to post-harvest, and pretty much across any flow event, the harvested basin's exporting a lot more nitrogen. And if we throw that into the mixed effects models, and this is a similar graph to what I presented before. This shows the post-harvest change in the 100% basin compared to pre, how much more it's exporting post-harvest. So we're looking at about a 5 kilogram per hectare increase in 100%, about 10 in the forest practices, and about 15 in the 0% harvest. Looking at total phosphorus. Total phosphorus is a little bit different. Where nitrate is very soluble, total phosphorus is usually associated with sediment, and it's almost always in much lower concentrations than nitrate. And in this case, if you look at pre-harvest concentration, in contrast to nitrate, TP concentrations some of them went up, some of them went down. There was no rhyme or reason to why. I mean, I couldn't figure out, there's really no pattern in why some went up, some went down, but they just varied quite a lot. And when we look at export, so in spite of some of them going up, some of them going down, I think it was just the increase in total flow is responsible for this generalized increase in total phosphorus export post-harvest. And again, you look at monthly patterns here. Again, it's just like sediment transport. The greater the flow, the more you're going to get transported out. Again, flows over here on the right axis, these black dots. But you don't see the big changes post-harvest. I think the increases here we saw were fairly small and, again, largely flow-driven. But when you sum them up over the year, the response among all three treatments was very similar. There's really no difference, statistical difference among the three of them. And finally, in conclusion, I would say turbidity and suspended sediment export are both very low pre and post-harvest. The vast majority of the turbidity measurements were low, where the error in predicting suspended sediment concentration, hence the model predictions are, the error in there is relatively high. So I don't think those, those estimates we came up with for total export are probably biased quite high. And we don't think there was really any meaningful change in export detected. And for nitrogen, again, pre-harvest variability in concentrations was much higher than we expected, but in spite of that, concentration increased post-harvest at all the sites. And it's the nitrogen export increased in all treatments, and that was also correlated with that increase in runoff and with the proportion of the basin or of that site that was harvested. And with total phosphorus, there was really a little change in concentration after harvest, and it was inconsistent among the sites. And the total phosphorus export did increase in all treatments, but that's probably a function more of increased runoff than anything else.